So, one component of the cell that bears going into in more detail is the membrane. In order to understand how certain hormones and cell communications and endocrine elements work, you have to understand how the plasma membrane works. This is something that's extremely important to understanding muscle function, nerve function, the endocrine system, all sorts of biology. To start off with, let's talk about the functions of the plasma membrane. So, the plasma membrane keeps the inside of the cell in and the outside of the cell out. To a certain extent, it does much the same thing for the cell that your skin does for you. But if the outside is out and the inside is in, and if that was an absolute barrier, then the cell would have no way of ever being able to know what was going on in its environment. So, just like your skin has in it sensors that can detect the outside world, it has your eyes, your nose, touch receptors in your skin, things like that. The cell membrane, or plasma membrane, as it is sometimes called, has within it receptors that can detect the outside world. So another part of its function is to communicate information from the outside world to the cell. The plasma membrane helps give the cell its shape. I mean, in sort of the same way that your skin gives you your shape. That makes sense. And all cells have to exchange material with the outside world. They have to get energy from someplace. That means they have to eat, they have to drink. So food and water have to be able to get into the cell, and they have to be able to release waste products. So the plasma membrane acts as the gatekeeper of the cell. It lets some things in, things that are supposed to get in, but it keeps other things out. This concept of letting some things in but keeping others out is known as selective permeability. The cell membrane has selective permeability that is based around several things, but the primary level of it is based around its nature and what it's made out of. The primary component of the cell membrane is phospholipids. We talked about phospholipids in the past, basically the way phospholipids work is they have a charged phosphate group on one end. Phosphate has several negative charges. It's very highly charged, very water soluble. And it has two fatty acids coming out of it. Fatty acids, of course, are uncharged and hydrophobic. So here we have our phospholipid. It has a hydrophilic side, a water-loving side, and it has a hydrophobic side, a water-hating side. If you take phospholipids and you just throw them into water, you're going to disrupt that water's connections. Remember that water wants to form as many charge connections as possible. So if you were to just have phospholipids randomly floating in the water, note that, so up here we have the charged end of the water molecule associating with the charged hydrophilic portion of the phospholipid. Here you have a good connection as well. But there are a lot more areas, because the tails are very long, where the water is not forming good connections. It's got its charged portions next to a hydrophilic structure, which it hates to do. Fortunately, there is an easy solution to this, and it is called the phospholipid bilayer structure. I'll show you the phospholipid bilayer structure in just a second. But first I want to talk about what lipids you're going to find in the membrane. So phospholipids are the major component. About 98% of the molecules are lipids. Of those lipids, 75% of them are phospholipids. Phospholipids are amphipathic, meaning that they have a hydrophilic side and a hydrophobic side. Hydrophilic phosphate head groups and hydrophobic fatty acid tails. About 20% of the lipids in the membrane are cholesterol. This is something that you find in animal cell membranes that you find a lot less of in plants. This is one reason why meat and animal fats like butter and lard are high in cholesterol, whereas plant fats like olive oil or vegetable oils tend to be low in cholesterol. The cholesterol's job is to stiffen the membrane. Because cholesterol has those ring structures, it has a very stiff, hard structure. Whereas the fatty acid tails of the phospholipids are very fluid. So the cholesterol will help stiffen and solidify the membrane, keep it from getting too fluid. The cell membrane is sometimes described as a semi-fluid membrane. It has some fluid characteristics, but some solid characteristics as well. About 5% of the membrane lipids are glycolipids. Now remember, anytime you see gly, that means sugar. So these are going to be lipids where you have two fatty acid tails going off on one end, but instead of a phosphate group, or actually attached to the phosphate group, you have carbohydrate chains. These carbohydrate chains can serve as markers, like for identification. We said that cells, if you'll recall, are labeled with carbohydrates so that your body can tell self from non-self. They also contribute to what's called the glycocalyx, which is just many cells have a sort of starchy, snot, mucusy covering over them that helps them stick to surfaces. This is the phospholipid bilayer. This is the solution for that hydrophobic problem. So you can see here, we have our phospholipids are arranged in such a way that their tails face each other. And their head groups all face the water. This allows water to constantly be in contact with other waters or charged hydrophilic things, like the head groups. All of the tails are facing other tails, so no water has to be in contact with the hydrophobic tails. Hydrophobic tails, maybe they're okay being in contact with other hydrophobic tails, but the important thing is that the water isn't in contact with anything hydrophobic. And so no unpleasant, no low or high energy interactions are forced in. But in this bilayer structure, like we draw it as if it's all one thing, but this is one phospholipid here, this is a different phospholipid here, this is a different phospholipid here. So they are, while they're all held into this configuration, they are not necessarily attached physically to each other, as long as you don't force any water into the middle. So if we take a look here, you can see the semi-fluid nature of the membrane. And you can see that the individual water molecules are capable of drifting side by side. Not water molecules, sorry. The individual membrane phospholipids. So they're fluid in a sense. Each molecule, each phospholipid, has lateral movement. It can move side to side, and remember, it's a membrane, so it's a two-dimensional structure. So they could also move, into and out of the screen or the picture that you see here. But they can't really move up or down. If they were to move up, it would expose these hydrophobic tails to the water. Water doesn't like that. If they were to, move down, same thing. So, while it's not impossible for them to move up and down, it takes a lot of energy. So it doesn't happen very often. Now, even though we said that 98% of the molecules in the membrane are lipids of one sort or another, it is not just lipids that float within the membrane. There are also proteins. So here we have a big protein, right? About 50% of the mass of the membrane is proteins. But wait, I hear you think. Didn't you just say that 98% of it was lipids? Well, there's a difference. I said 98% of the molecules by number. If you were to just count the molecules, 98 out of 100 of them would be lipids. If you were to weigh the molecules, there's about an equal amount of weight of proteins and lipids. What does that tell you? It tells you that proteins are a lot bigger than lipids. They're like huge ships floating in a sea of water, right? The water molecules are very small, the ships are huge, water moves quickly, the ships move relatively slowly, but one ship is the equal of a lot of water. So we have proteins floating within the membrane as well. We have membrane lipids flowing back and forth, and proteins can also float side to side, floating within this semi-fluid membrane. There are two types or two general classes of proteins associated with the membrane. Integral membrane proteins are proteins that are actually stuck in to the membrane. They go at least part of the way into the membrane, and most integral membrane proteins are what we call transmembrane proteins, meaning that they go all the way across the membrane. Integral membrane proteins are stuck there. There's really no way to get them out of the membrane without disrupting or destroying the membrane. You also have peripheral membrane proteins, which are not so much in the membrane as on the membrane. They're usually just sticking to the membrane, like sticking to one surface of it, often by charge-charge interactions. So like the membrane is negatively charged, so maybe this protein has like some positively charged portions here that are going to stick to it. Peripheral membrane proteins can fall off of the membrane and come back onto the membrane without disrupting the membrane itself. You don't have to do anything bad to the membrane to get them to fall off or fall back on. In fact, you can usually do it by exposing the membrane to high salt levels. Here's a depiction of the membrane with all of its various components that you might see in it. So here we see most of it is membrane lipids, right? So like here where I've circled, you can see the phosphate head groups, and you see the tails going into the membrane. These blue things here, those are cholesterol, which are stiffening the membranes. These chains here, those are the the sugars attached to the glycolipids. Here we can see a transmembrane integral membrane protein. And there are several different types of integral membrane proteins, which we'll talk about in a bit. But one of the main types is what's called a channel. A channel is a protein that exists to let things move through and into the cell. And they're very specific. Like, they aren't just an open hole. They only allow certain things through. You can see here these peripheral proteins stuck to the surface of the cell. Note that outside of the cell, the glycolipids all face outside of the cell. That's so that the cell can be identified, right? You don't need glycolipids facing the inside of the cell. The cell already knows that the inside is part of the cell. But inside the cell, you have these cytoskeletal elements. These are microfilaments, mostly, that if you'll recall from the last lecture, I said that microfilaments are what determines the shape of the surface of the cell. And they do that by actually hooking in, attaching to the proteins that are found in the membrane. What sorts of proteins are you going to find in the membrane? Well, there are several different types, but two of the most important are going to be channel or pores and receptors. Both of these exist to move things across the membrane, but they're moving different things across the membrane. Channels allow physical, chemical molecules to move across the membrane. So, for instance, this is a special type of channel protein called an aquaporin. And so these are waters out here, and you can see this aquaporin is going to allow water molecules to move across the membrane. They're going to move through this channel, so they never actually come in contact with the hydrophobic tails. Receptors, on the other hand, they exist to move information across the membrane. So receptors have an external portion that binds a special molecule called a ligand. The ligand binds to the receptor, and that causes a conformational change in the receptor that sends a signal across. Note that the ligand never actually moves across the membrane, but the information that the ligand exists does, and therefore the cell can react to the presence of the ligand, even though the ligand never actually enters the cell. So, the types of proteins found in the membrane. Receptors and channels or pores are two of them. I want to let you know like what the other types are and throw a little bit of detail on it. So receptors are one of them. Receptors, again, bind to a ligand or a chemical messenger, and then they produce a change inside of the cell that is detectable. Some of the proteins inside of the membrane are enzymes, so they may bind to something and then chemically change that thing. Some of these enzymes are going to actually be on the outside of the cell, but more of them are going to be on the inside of the cell. And often, enzymes and receptors, an enzyme may function as a receptor. It may bind a chemical messenger on the outside, and then that may activate an enzymatic activity inside, and that enzymatic activity is what is actually detected, seen by the cell. There are channels. Channels let things pass through. Often, but not always, ions. As I said, there's a special type of channel, the aquaporin, that allows water through. Some channels are always open, right? So for instance, aquaporins are always open. They always allow things to pass through, and they can pass through in both directions. Some channels are what are called gated channels. Gated channels are like a door. The door can be open or the door can be closed. When the door is open, things can walk through. When the door is closed, it's locked. They can't. So these are channels that can be opened or closed. And there are cell identity markers. These are what are called glycoproteins. Just like glycolipids had sugar groups attached to a lipid, here we have sugar groups attached to a protein, and they serve much the same function. They are recognized, a part of your body's ability to recognize self versus non-self cells. And last, we have cell adhesion molecules. These are going to be cell surface proteins that stick to something. Usually, they are either going to stick to a similar protein in another cell, and this is what allows two cells to bind together. If your cells couldn't bind together, you'd just be a big massy blob of like liquid cellular tissue on the ground. Your cells have to be able to bind each other in order to form three-dimensional structures like organs. And they can bind either other cells, or they can bind just proteins that happen to be, moving along through the outside here, and they might just bind onto the protein. The protein is a part of the extracellular matrix, which is a concept that we'll discuss in more detail later on. All right, so to reiterate what you need to know for this lecture, you need to know what a phospholipid is and what it means for a phospholipid to be amphipathic. You need to know what a phospholipid bilayer is and why phospholipids arrange themselves in bilayers in the membrane. You need to understand the concept of semi-fluidity and selective permeability. You need to know the two classes of proteins, integral versus peripheral, and you need to know the 6 types of proteins based on their function that you will find in a membrane.